Let me tell you a little bit about SARS-CoV-2 entry. And this will tell you why hydroxychloroquine could never work. You guys remember hydroxychloroquine? The fiasco that was, okay? So SARS-CoV-2 has a spike protein here. It binds to ACE2. The fusion peptide is buried down here, this cyan-colored sequence. That's the fusion peptide. And for fusion, which is not triggered by low pH, it's triggered by cleavage of the spike. The fusion peptide is thrown up here, and then it can insert into the membrane and catalyze fusion. So the fusion of this spike, or the reorientation, is caused by protease cleavage. And in the lung, there is a surface protease called TMPRSS2, TMPRSS2, which when the virus binds ACE2, the protease cleaves the spike, it reorients, and the fusion peptide inserts in the membrane, and it fuses, and the RNA comes right in at the plasma membrane, okay? So in the respiratory tract, that's how the virus gets in. In other cell types that don't have TMPRSS2 at the surface, the virus is taken up by endocytosis. The virus ends up in the endosome, and it binds to a protease in the endosome called cathepsin L, which cleaves the spike, and therefore the virus can fuse from within the endosome. That doesn't happen in the lung. It happens within any other kind of cell type that the virus might encounter. The cleavage in the endosome by cathepsin L requires low pH to activate that protease. Not anything to do with the virus. The low pH activates the protease, and hydroxychloroquine inhibits the acidification of endosomes and therefore inhibits, endosomal entry. Now, a group of scientists, they weren't virologists, when COVID emerged, everybody wanted to work on it because they could get money. Pigs to the trough, my colleagues called it. No, sorry, they're not pigs, but you get the. And a group in France took SARS-CoV-2 and infected kidney cells with the virus, where there's no TMPRSS2 on the surface. Why did they do that? Because they didn't know anything about virology, okay? And hydroxychloroquine inhibited that infection in kidney cells because it's inhibiting the protease that cleaves the spike in the endosome. So this was spread far and wide in the journals, and the U.S. president at the time forced the FDA to approve this without any kind of clinical trial. It's the worst period in FDA history ever. And this didn't work because it doesn't inhibit reproduction in the lung. It can't. If you know the biology of the bloody virus, you know it won't work. And they should have done clinical trials, but they didn't. People actually died of hydroxychloroquine treatment because it has other toxicities. So the FDA rapidly revoked its emergency use status. But to this day, you will find people who want hydroxychloroquine. You will find hospitals in the US that give it routinely to COVID patients along with antibiotics. Yes? Same idea. Cell culture experiment showed it could inhibit virus reproduction. But no clinical trial ever demonstrated a benefit of ivermectin. And yet still, to this day, people want it. And you can buy veterinary ivermectin to treat your animals, and people find it. And they take it, and it's bad because it's a different concentration, and it can really hurt you. We call it horse paste. Give it to your horses for deworming. So that's a really interesting story of how you shouldn't let politics interfere with the science. This is why hydroxychloroquine could never work. And don't argue with me, because that's the biology. I'm not making this up, folks, okay? People want to argue with me all the time. Oh, you're just getting money from pharma. I wish I was. I'm not. This is the science. I go by the science. Class 2 fusion protein